All right, folks, we have arrived at the grand finale of the talks. And for our last talk, we have Krishna Manoj from the lab of Domitilla Del Vecchio at MIT, who will tell us all about synthetic biology.
Hello, everyone. Thank you for the introduction, and thank you to the organizers as well as NITMB for organizing such a great workshop. And it was great to meet such amazing set of people. All right, so as he said, I am working with Professor Domitilla Del Vecchio, and I'm here on behalf of her. And today I'll be talking to you about how modularity was challenged in synthetic biology for the last 20 years or so. So the outline of this talk is going to be as follows. I'm going to start off with the vision of programming cells to behave like machines and how this vision was challenged by context. Specifically, I'll be looking at retroactivity, cellular resource sharing, and ectopic resource sharing or synthetic resource sharing. And in this segment, I'd be showing you how context-dependent affected modularity and what we did to tackle this context dependence to regain the modularity. And finally, once we've regained modularity, I'll end it with where do we go from here. All right, so it's starting things off with the vision. So modularity is this key abstraction that lets you essentially reduce the complexity while designing complex networks. So for example, if you want to create a computer network, you can do a layered approach where you compose multiple modules together and just have it work. This essentially gives you a certain advantage where you can forget the inner details of a module and only focus on the input-output connectivity when you're composing this huge layered network. And usually in computer networks, feedback is used to maintain this input-output property irrespective of where you put this. So now this was an amazing thing, and we were really tempted to apply this in biology, and that's where synthetic biology comes in picture. The vision was to essentially use the same approach of modular layered composition design in biology. And this was supported by the fact that we had a large library of genetic paths, as well as an ability to synthesize DNA. So this was like, okay, this vision is there. We can do it. And we took this vision to an extent where we have automated softwares like Cello and Comet and many others, in which it's like it leverages modular composition and it designs the genetic networks for you. And it came into fruition where Tay-Sach Moon had a 11-gene network which was a 3 AND gate network, which performed the way it had to. But the thing is, for even a 11-gene network, it took 5 years in a controlled lab setting to make it, even though the automation software designed it for you to begin with. So why is this the case? Even though you have a network already designed, why is it so hard to implement it in biology? And for this question, when you actually design a genetic device in biology, most of the time when you place it in a biological setting, the dynamics is unpredictable. And most often, this unpredictability comes from the fact that there are indirect connections, not necessarily the direct connections you intended. And this, because of these two reasons, what happens is that whenever you design a new component into a biological system, you have to redesign your entire network to make sure it behaves the way you want it to be. This is where this analogy between a computer network and a biological network fails. In computer networks, this modularity can be maintained by adding negative feedback or just by making sure the two components are physically separated. And this kind of insulation is for a large extent missing in biology. In biology, whenever you add a module or add a genetic device in a cell, context plays a role. Context as a whole can be broadly classified into 3 high-level classifications. I'm going to tell you what this means. If you're interested in designing a genetic module, the first thing you have to look at is the genetic context. You're placing this gene into a system. You have to see what the position is where you added it, what your surrounding genes are, what the directions of the surrounding genes are, what's the chromatin state. So all of these are context in a genetic level. Now you zoom out, you have cellular context, you have off-target bindings, you have resource sharing for like common cellular resources, you have growth rates which would affect things, you have retroactivity or intermodule dependence. That's a cellular context. You zoom out even further, you have the environment itself, the pH, the nutrients, the temperature, or intercellular interaction. So all of these are the context which affects the performance of the genetic module which you designed. In this talk, I'll be focusing on the cellular context, specifically on resource sharing and intermodule loading. So this brings me to the second portion of my talk. So in this section, this is how I'm going to go. I'm going to start with telling you what these are. I'm going to tell you how it shows up in a biological context and what we did to tackle this to regain modularity. For each of these, I'll be going through this exact same pattern. All right. So starting with what each of these are. Let's say we want to design a genetic module with input U and output Y. And we've characterized it as with a Y versus U plot as shown here. Now, if you add a downstream system to it, the downstream system could affect the output transmitted by the input system, the output transmitted by the upstream system. And this is what is called retroactivity. So here you can see the isolated system, which is shown in black, is considerably different than what happened when the system was connected. Next, if you are looking at combining two modules, for example, here we have two activation cascades being combined, you expect the system to show an activation cascades behavior. But because both of these modules would use the same cellular resource, and the cellular resources are usually limited, there's going to be some coupling between them and emergent behavior, which is going to change your actual behavior. All right. And finally, when you have a lot of modules competing for the same resource, as you add input to one of the modules, the efficiency of another module would change, showing that there's going to be coupling between these modules induced just because of these resource sharing, right? So these are some quick examples and just showing you what retroactivity and how resource sharing are going to be affected. I'm just going to go into detail into all of these. So starting with retroactivity. So as I said before, retroactivity is when you have an upstream system, when connected to a downstream system, has a back action or a backward reaction flux from the downstream to the upstream, causing a change in the output of the upstream system. An example of this would be, let's say you're looking at a genetic clock. Such a genetic clock in isolation would show sustained oscillations. But if you connect this genetic clock to an extra molecule, just so that it just uses a little bit of your activator, you can see that the amount of activator is lower for your original genetic clock, causing a difference in your original system, thereby making the oscillations completely disappear. This is what is called retroactivity, and it's not uncommon. It's there in other physical systems. For example, if you look at an electrical system, when you connect an extra impedance, the voltage would drop, showing that there's a retroactive effect when you have a downstream system connected. A similar thing can be inferred from a fluidic system, where when you attach another fluidic chamber, your pressure would drop, and therefore the flow in which the fluid flows from one chamber to the other would change when connected when compared to in isolation. To go into a little more detail in our biological setting, there's a little bit of math. So let's consider an upstream system, which is just a transcriptional module. You have U being the production rate of your particular protein and Y being the molecule of interest. The ODE, which describes the change in Y, would have the production rate minus the decay rate. When you add a downstream system, which is just a simple promoter in which this molecule can bind, you have an extra reaction, which is the binding reaction, unbinding and binding for that particular molecule. But you have the extra term which comes in your original Y, the ODE for the Y molecule. This is what is retroactivity, and this is the reason why the output of your upstream system changes due to the addition of the downstream molecule. Numerical simulations of such system has shown that if you have a step response, the connected system shows a slowed down response when compared to your output, when compared to your isolated system. And when you look at an input which is a sinusoidal wave, we see that the amplitude of the connected system is lower, showing that we have a lower bandwidth due to retroactivity when the system is connected. We have performed experiments in bacteria, in vitro, and in yeast. And here I'll be showing the experiments we've done in yeast, which shows good similarity with our predicted model results. All right. So now here we've looked at a single upstream system and a single downstream. We could have more upstreams and more downstreams. So to generalize this a little bit, let's say you have two upstream systems and a single downstream system. And we're interested in looking at what happens if you apply a perturbation to one of the upstream systems. When you just think about it, if I increase X1, what will happen is X1 is going to go and bind to X3, releasing more X2. So X2 would increase. So this is essentially when X1 displaces X2 from X3, if X3 is a DNA, thereby increasing X2, one form of retroactivity. Another case would be if you have a single upstream system and two downstream system. If you increase X3, X3 is going to use more resources from X1, thereby limiting the amount of resources available for X4, thereby dropping X4. So this shows that sequestration of X1 by X3 causes X4 to drop. In a similar manner, what would happen if you have a huge gene network? You have multiple retroactivities, which is going to interlink all these connections. Yes.
Can you clarify the meaning of these edges from X1 to X3? What does it really mean if there's an edge?
In this one, it's just X1. Okay, this is a very simple schematic. It's not really an interaction graph. It's more like X1 interacts with X3. That's about it. It's not like it's activating, it's repressing, nothing like that.
Okay.
Yeah. Okay, so that's a big question. I'm not going to answer that question because that would take a lot of time. And if there is a semi-answer provided in the paper over there, today what I'm going to show you is I'm going to show you a simpler network, which is very common in mammalian systems, where you have an activator and a repressor targeting a target gene of interest, but these regulators also have multiple binding sites in the genome, which you're going to model as decoy sites. What we've seen is that in the low activation case, you have low amount of activation or basal expression, but as you increase the amount of activator, what happens is the activator goes and binds to the decoy sites, which releases the repressor, which represses the target. So essentially, if you look at the target expression with respect to the activator, you're seeing repression. We've designed this genetic circuit in bacteria and where the activator was CRISPR-A and the repressor was CRISPR-I, and we were able to obtain this experimentally. So here, in isolation, the activator behaved like an activator. It activated the system. But when you put the rest of the system in there, the activator essentially became a repressor. So this is essentially retroactivity. And retroactivity is, of course, bad. We don't want a module to essentially change its behavior when we add other things to the system. So how do we deal with this? A way to tackle this retroactivity is by designing insulation devices. So here's how an insulation device works. So this was our original problem. You have an upstream system, downstream system, and retroactivity going in there. We essentially want to separate this upstream and downstream system and add an extra system inside such that the retroactivity which comes from the downstream system is attenuated, and the insulation device itself will give a retroactivity which is approximately 0 to your upstream system, thereby making sure that the signal which is transmitted by your upstream system is retained when you're connecting to a downstream system. So the way this works is, so now it's like you're essentially attenuating S. This is a simple controls robustness problem where if you think that this is your insulation device, you have your input U, you have your output Y, retroactivity comes in, you're designing this negative feedback to make sure that in a connected system, your Y is independent of S. the math looks like this, where as you increase y, the input which goes into the system is lower because you have a negative y term in there, thereby compensating for the original increase in y. And at high gains, you are essentially getting a system which is independent of the loads or retroactivity in this case. Such a system can be implemented in biology using fast covalent modification cycles. And we've incorporated this in the yeast system, which I showed before. So here was the experimental results, which I had shown before in yeast, where due to retroactivity, we had a smaller amplitude. Now, adding the insulation device, we were able to get the amplitude back, and the isolated system followed the connected system in a really good manner. So using this insulation device, we were able to ensure that the signal was transmitted from your upstream to the downstream system without any back action / problem to the upstream system. All right. So this concludes my section on retroactivity. Now I'd show you what happens in the case of endogenous resource sharing or sharing for like cellular resources. In this section, I would start with two examples where systems fail due to this resource sharing. To start things off, we have a multiplex sensor. We have two sensors here. The first sensor looking at input U1 and giving us an output Y1. The second sensor sensing the input U2 and giving us an output Y2. In isolation, you can look at the first three portions of this graph. They work very well. When the input is high, you have a high output. And therefore, we expect that when both inputs are present, we are able to sense both of them. But in reality, we see almost a 60% drop in the response. And this is because both of these sensors use the same cellular resource, which was limited, making the final output lower than what it was supposed to be. The next example is an example of a genetic logic gate. For example, here we are designing an AND gate with two inputs U1 and U2, and we are interested in the output. The desired outcome is that for the case where you don't have either input or you don't have any of the input, you have a low response. And when you have both the inputs, you have a high response, just like how our AND gate should behave. But in experiments, we see that that is not the case. The behavior is completely flipped when you actually implement this circuit experimentally. And this is because when you design an AND gate, you have cellular resources which are being shared by both of these modules. But because of preference between the resources to one of the modules, you have an expected change, which makes your experimental results or actual behavior change from the expected outcomes. A way to see what is happening in this case is to look at interaction graphs. So let's say you have a few nodes. Due to resource competition, if your first node is an activator, it essentially becomes an effective repressor to all the other nodes with which it's not connected to. And similarly, if it was a repressor, it's essentially an effective activator to all the other nodes. Doing this to our specific system, we have an activation cascade where the expected dynamics was, of course, activation. But because U is right now an effective repressor to X2, we have an incoherent feed-forward loop. And for an incoherent feed-forward loop, your dynamics would be a pulsating dynamics where you have a peak for an intermediate value of U. And so this experiment behavior is in line with the expected behavior with the network, which includes this hidden interaction. Now, the question is, how do we make sure this doesn't happen? How do we have our network behave irrespective of resource competition? So the way we deal with this is resource competition can be thought of as a disturbance in your available resources. So you can frame it in a control theory framework where you have a genetic module, which has our output protein, and the disturbance coming into the genetic module due to resource competition. To make sure our circuit is robust to these disturbances, we have two approaches. The first approach is designing a negative feedback, which will sense the current output and compensate for the disturbance. Or we could have a feed-forward approach, which will sense the disturbance itself and pre-compensate for the output. In today's talk, I'd be focusing on the first approach of designing a negative feedback control and mainly using this. Okay, before I go there. Okay, this is the experimental circuit where you have a genetic module which produces GFP. Your competitor is something which produces RFP. So as you induce the RFP production, so you can see from time 0, RFP production increases. This causes resource competition, which is shown as GFP going down. Now we are adding an antithetic motif, which was shown from the Mustafa Kamash's group, where you have a short sRNA, which is activated by the production of the protein, and it degrades the mRNA, such that therefore creating this negative feedback loop system. And experimentally, we see that we were able to, in the same exact setup as before, we've induced RFP, and we were able to see that GFP was almost a constant in time, thereby reducing the effect of resource competition in the system. All right, so here, with this integral controller, we were able to make sure to get the modularity back, where your GFP module was able to perform as before, even though there was competition going on. Okay, and the last part is ectopic resources. This is a little bit more trickier, because when I say ectopic resources, these are synthetic resources. These are things which we add in the cell. So if there was competition, we could always just add more of these. So the question is, why is this a problem? So for ectopic resources, the problem comes when these resources themselves are toxic to the cell and therefore limited. So there's a limit to which you can add these resources. And because of that limit, you get competition effects. I'll be showing this with a case study of CRISPR, where for CRISPR, you have these dCas9 and an activator protein, both of which are toxic to the cell. So here we have both these ectopic resources, dCas9 and an activator protein, and multiple CRISPR modules. I'm just going to quickly go through what inputs and outputs mean in this scenario and how this fits into our resource competition framework. For CRISPR, you essentially have this guide RNA, which is this tiny squiggly line here. You design these guide RNAs such that they'll go and target specific spots on the genome. So anything about the genome which the CRISPR complex targets is completely encoded on the guide RNA. So you can say that all these inputs are your guide RNAs, and you can design multiple such guide RNAs to target multiple such genomes at the same time. And then both dCas9 and the activator protein are resources which are shared by all of these RNA molecules. All right. So first, let's look at CRISPR-I, which is repression using CRISPR. In CRISPR-I, you have a guide RNA which combines with the dCas9 molecule, forming a complex, and this complex goes and sits on top of your promoter, essentially being an obstacle for RNA polymerase to come and bind. And that's how this CRISPR-I works. That's how CRISPR represses protein production. Here, if you have two CRISPR-I modules, with first one having an input U1 repressing RFP, and the second one having an input U2 repressing GFP, we see that as you increase the amount of U2, which is the input for GFP repression happens as expected, but there is de-repression of RFP, which was not expected. Experimentally implementing this, we were able to see that as you increase the amount of competitor, we were able to see a 30-fold increase in the RFP production. Now, in a similar manner, when we look at CRISPR-A, oh my goodness. Okay. In CRISPR-A, sorry, this happens if you put things in Dropbox, the pictures just change into another picture. But in CRISPR-A, you can take my word for this, these two molecules essentially bind together. So, and then this activator protein is like a magnet, which increases the amount of RNA polymerase, which comes to the area of the promoter, and that's how it increases the production of the activator protein. But the funny part with CRISPR-A is that because you have these two proteins which have to bind to the sRNA, the amount of reactions or combinations which could happen is more than what you have in CRISPR-I. In fact, you have this diamond architecture or reaction network which happens just because you have two proteins in there. So the consequence of such a reaction network is that, this guy is so bad. Okay, so of the reaction network is that as you increase, even if you just have a single module, as you increase the input, due to internal competitions, you get a biphasic response. This is similar to the hook effect or self-sequestration and other effects which has been shown in other T cells and other systems. And we were able to show this experimentally as well, where when implementing such a system in bacteria, we were able to see a 90% drop in the response of our system. Now, looking at two modules, the picture's here. Okay, when you look at two modules with CRISPR-A for module 1 with input U1 and output RFP, CRISPR-A module 2 with input U2 and output GFP, the model predicts that as you increase the input U2, GFP increases and the activation was reduced due to the coupling between the modules. And we were able to show this experimentally as well, showing a 60% drop in the activation efficiency of our CRISPR-A module. The next thing we did is, now that we know competition exists, and we know that these are synthetic proteins, we can actually add controllers directly on these proteins. They are not on the chromosome, they are on plasmids. So we designed negative feedback controllers to make sure these protein levels are maintained at a constant rate. So this is how we designed them. For example, here you have the regulated Cas9 generator. So this is the genetic module which produces Cas9. We added an extra guide RNA which would target the Cas9 promoter. So the guide RNA will sense the amount of Cas9 and negatively regulate the Cas9 production. So if you have an increase in Cas9 production, the feedback will repress, thereby reducing the production. And if the Cas9 was lower, the feedback would de-repress, increasing the production of Cas9. That's how the negative feedback regulation works for the Cas9 generator. In a similar manner, we designed another one for the RBPAD as well. This is a little bit more complicated because RBPAD is an activator. So you won't be able to make a single step negative repression. So we added an extra step in which the RBPAD activated a repressor, which then repressed RBPAD. So now we've designed this, we've checked this in our model, it works. Let's see if it works experimentally. When we implemented, this is our earlier system where we had an unregulated Cas9 generator for CRISPR-I, which showed the 30-fold increase. Implementing our Cas9 controller, we were able to get a pretty good response where, irrespective of the amount of competitor you added, we were able to maintain the repression in RFP. For the case of CRISPR-A, earlier we had a 60% drop in activation. Adding the Cas9 regulator, we were able to reduce the activation drop to 30%. And when we added both the regulators, we were able to get the drop to almost a non-significant level. So with this, essentially, this brings me to the end of our second thing, where we've looked at three different types of retroactivity, or three different types of context dependence, namely retroactivity and cellular resource sharing. And we've looked at how we design stuff or design controllers to ensure that modularity was regained. Now, where do we go from here? Now, we essentially went from this original vision, which was 20 years back, to a new vision, which incorporates context dependence. This would imply that when you're designing a circuit, you know context dependence is going to come in. You could either design a controller a priori to make sure context dependence doesn't come in, or you can make a model which includes context dependence. And that's ways to make sure that your system behaves the way you want it to, even though context exists. So to conclude, we know that modularity is amazing because this implies that we can layer stuff and make complicated circuits. But modularity as it's intended is often missing in biology because of context. There were two solutions which were proposed. One, you have a design approach which makes sure, make a model which includes context dependence in there. The question is, okay, do you need to make a new model each time a new species gets added? How many new models do you have to make for such a system? And another question is there any way we can use or modify or AI or ML-based models to ensure that these unknown interactions can be accounted for while looking at modular design? The second solution was to make a controller to ensure that things are regulated. But then, is this scalable? How many controllers do you make? How do you decide which genes get controller and which doesn't? Yeah, so these are the possible solutions and possible questions we would like to get answers to. So with this, I would like to acknowledge all the current members of our lab and all the past members who've contributed to most of the work which I've shown here, and our funding agencies.
Thank you. Thank you very much.
Yeah, I thought that was a great talk you gave there. It was really interesting to see you are approaching it from, I want to engineer a system that has modularity, whereas we're sort of approaching it from, we have a system, are they modular? And the first thing I noticed is you're thinking about shared resources. In the modular construct in signaling pathways, each module, as we identify, has unique low concentration components. They share components, but all the components that are shared between the modules of the signaling pathway are present in very high excess in the cells. So that actually there is no such thing as resource competition in a normal signaling pathway. But what's interesting is that some of the proteins that are shared between them, if you change the expression level, you get this indeterminate physiological effect on the cell that people haven't been able to figure out. And we sort of propose it's because of what you say. So the point is that evolved systems understand what you've shown here, and they've sort of evolved solutions that avoid those things. And so understanding these things, I think, is really important to identify natural modularity in cells. So really nice job.
I wonder, I mean, this is a gene regulatory network, I suppose, right? So did you try to find whether in some real biological system you find those controllers, similar controller patterns? So that's well, I'm just curious, does evolution actually, find this solution to separate the modules? And if you find many of those, whether you can learn from the evolution, that's how can you scalable this, right? Make this scalable, adding more controllers, and how the evolution actually resolve that. That might be, yeah, I'm not sure if you have done those.
I think we have not done that explicitly, but it was a happy coincidence because right now we have a research going on, which is on chaperone competition, like for folding. Interestingly, there is a negative feedback which regulates chaperone levels already existing in the cell. It is not a high gain negative feedback, it is low gain, but I think from the results we've seen, I think it's the low gain is enough to maintain those levels in the cellular context.
It doesn't have to be perfect.
Exactly. So I think, okay, this might be my perspective, but I do think that cells have evolved such that for whatever competition they do actually see in there are feedbacks, not necessarily as high again as what we have made in the synthetic.
I just wanted to swear.
Yeah, it's fine.
And then wait 1 second. Done.
Okay. I kind of building off of Steve Wiley's point. I'm trying to understand if you have a low amount of some, let's say it's a protein, then you're going to have retroactivity in the sense that you have a downstream effect, but then that's going to suck away that protein, and then it's not going to be able to do whatever was upstream as much. But if you have a whole bunch of that protein, like really high concentration, then you're going to run into resource sharing issues. So is that correct?
Mm-hmm.
So it seems like whether you're on the high end or low end of expression, you have retroactivity either way. Is that accurate?
Yes, that is true.
Interesting. And then the insulators, I understood how the insulator was able to insulate against retroactivity. Was that a different design that you were able to implement, or is it kind of for the case of resource sharing compared to insulating retroactivity, or is it kind of like the same overall design principle?
That's a good question. So this kind of brings like to a bigger discussion whether retroactivity and resource sharing is actually two different things or it can be clubbed into a same thing. I'm not going to comment on that, but I think a huge consensus is that it's kind of similar. It's just cellular resources can be looked at as an upstream system and a downstream system. But to answer your question, in the talk, I showed that for retroactivity, we have the insulation device, which is essentially you're adding a new module in there, but which just make sure that, which is essentially interacting with both the upstream as well as the downstream system.
I see.
In the cellular resource case, when we added the antithetic motif, it doesn't interact with the cellular resource. So the original resource which was being competed for was ribosomes, and the antithetic motif is essentially an RNA. So it doesn't really use the ribosome itself. So it's like these are essentially three different ways in which you can implement something to get modularity back. But the way you implement it, it depends on what your final goal is, which is the molecule you want to make robust to perturbations.
Very interesting. Thank you.
Just a quick comment for Song and you. I'm just speculating here, but your controllers are quite expensive. Whereas in nature, I mean, they still have an issue at the gene regulatory level, but at the metabolic level, they're really cheap. So they're just allosteric effectors. So it may have got round. I mean, they could have evolved a protein, right, that the small molecule binds to, and then that protein binds to the enzyme. They never didn't do that.
That is true.
It binds directly to the enzyme that's responsible. So it's a cheap way of adding controllers.
Yeah.
But you don't have much option here at the genetic level. But these Cas9 things are expensive. So that's another issue,
Yeah.
But anyway.
Can I rephrase his question, but not in terms of expensive or cheap? How simple do you think these insulators would be in natural cells compared to your engineered cells?
I think insulators, because it's just phosphorylation cycles, they actually exist in natural systems. But if they're used for this particular purpose or not is not something I know of.
Oh, yeah.
Actually, you asked my question.
Oh, I did?
Okay. Yes, you did.
Oh, sorry. Damn.
No,
A good thing or a bad thing? So it's a little bit of a general question, but also a comment. So I feel that you're hitting into a difference in definition of modularity between engineering and biology in some ways. Like you're making your modules intentionally context independent so that you can build complex structure thinking as an engineer, right? Building blocks. But natural selection doesn't think that way, right? And modules in that case can be context dependent in biology, and they still are modules? Or is it that you find an abundance of context independent subsystems that nature prefers? It's a little bit broad, I know, but I thought I wanted to throw that distinction because I think it's key to what we were discussing from the beginning.
That is true. From a synthetic biology perspective, it's easy to say, yeah, that's the module. But from a general systems, like when you look at a huge network, like what was shown in all the talks before, it's really hard to say what's a module and what's not a module. And we've had things where like, yeah, this has a negative feedback. That would mean that this is robust. Can we call that a module? It's a bigger philosophical question than what I can give an answer.
Thank you for bringing us back to the fundamental conundrum that brings us all here. What is a module?
So my comment goes off exactly on this topic, right? Because we've seen multiple talks with respect to that the natural, quote unquote, natural modules are exactly context specific, right? So I understand, right, from an engineering perspective that you would want something that you can swap and play. On the other hand, you know, Steve gave us an example of a conserved functional unit. Let me not say a module, but a conserved functional unit that gets reused for whatever that function happens to be in multiple circumstances, right? So let's just leave aside for a second the concept of modularity, but looking for these types of heavily conserved pathway structures that serve a reusable purpose, right, in different contexts, right? Because talking to Steve, right, what does EGFR do? It tells you what the cell is doing, what sort of environment the cell happens to be in, and then processes that information into some sort of state change that occurs within that particular cell, which is agnostic to what the thing is on the outside and agnostic to what the function is on the inside. So thinking in terms of the functional requirements that cell has to do the things that it does, and then mapping that to conserved molecular pathway structures, right? Then I think that's a way to kind of get at what you're trying to engineer, right? Because those are parts that get reused for a particular purpose over and over and over again, right? So I think that's kind of, and it carries with it all the things from evolved systems, right? Energy efficiency or all that kind of other stuff, right? Within to that particular operationalization of that functional unit.
One more question. So a little bit to connect back from, you have a bunch of modules that work in isolation. And when you connect them, if you don't do anything, they're going to not work the way you want to. And one option, which is what you took, is to design them such that they are insulated from these effects. Has anybody tried to just put them together as they are and then use artificial selection to see if the cell can evolve the function that you want the combined modular but not optimally modular structure to do? And in other words, does evolution come up with similar solutions if you direct it to do the job that you want?
That's a good question.
Has anybody done that is my question.
As far as I know, no, but my literature might be very limited. But that is a really good question, especially given that we are synthetically adding these things and you want us to look at these systems in an evolutionary time scale.
Because you can't really avoid it because these are usually bacteria. So they're going to evolve really, really fast to get rid of your systems in any case.
To get rid of that
yes.
system,
So if you force them, if you do selection on them to do the job that you're asking for, I wonder what they come up with that you haven't engineered. That could give you some interesting insights into the problem.
You think the cells would want? The cells doesn't know what we want.
That's why you need the artificial. But that's why you need the artificial selection. You need a process by which you kill everything that doesn't do what you want,
or
eliminate some fraction of the things that don't do the job that you designed. So you'd have to kind of put them through some type of filter every generation or two.
Yeah, I think that's a really interesting project. I don't know anybody who's done that. And I think getting that artificial selection such that the cells does the process we want it to is quite difficult.
You have a reporter, you can sort them, no?
Oh, yeah, that's a great idea.
It depends. I don't know if you can sort bacteria, actually. I'm not an experimentalist enough for that. But
the sorting would require some cells to behave this way.
Yes, that's right.
That's a thing. So for having some cells to behave that way, we need that artificial something to make sure the cells evolve to do the thing which we want it to. And I think that is a little difficult to design experimentally. But that is a very interesting research topic.
Can I try building on? Like your GFP and RFP.
Oh, yeah.
Sorry. I mean, there's the idea that you could artificially do it. I mean, okay, again, not an experimentalist. So let's say if you don't have too many bacteria and coming back to your GFP, RFP, couldn't you just like, oh, well, the ones that are showing lower expression of whatever it is, I'm just gonna like remove them from the population. Is that doable in any way?
Yes.
It is doable, yes.
So then you're like manually selecting, right? So it's evolution, but you're manually removing them from your population.
Yes, that is possible, but yeah.
Okay, any other questions, comments?
Just to add on to that directed evolution idea, I think based on what I know about evolutionary genetics, like population size is really important for the evolution of these things. I don't know how many of you are familiar with Michael Lynch's idea about eukaryotic complexity arguably being much higher than prokaryotic complexity because effective population sizes have been much lower. Historically speaking, for eukaryotes than prokaryotes. There's billions of bacteria that might somehow exchange genetic information and be selected. And so when you say it speeds up evolution, it kind of depends what you mean by that. If there's higher selection, there's someone ready to replace you at any point, then it gets really granular, but you don't see as much like big jumps in complexity as you do with a lower population size. That's my understanding of it anyway. So I feel like if you were to do something like that, I feel like effective population size of bacteria or whatever you're selecting for would be really important. Just a thought.
All right. Let's thank Manoj again.